And I'm going to move through this relatively quickly. Here's what I want you to understand. There are some things that you will absolutely need to know and be able to do, but there are multiple layers of complexity here. There's a bunch of molecules and a bunch of processes that you don't really need to know or even be able to recognize. And the most confusing thing about everything that we're going to be doing today is that we're going to be looking at the same information stored in 1, 2, 3, 4 different ways. And this is how it actually works in living organisms. Information to make proteins is stored in 4 different ways. 2 of those look the same. 2 of those look the same. Everything is like mirror image. We're going to do this flip-flop, flip-flop, flip-flop thing. And remembering which is the flip and which is the flop is not nearly as important as understanding that if we start with the assumption that the molecules that we're looking at are chemically and physically built in such a way that information can be transmitted, you're good. That's the key piece. Information is conveyed through molecular structure. So we're going to be talking about DNA transcription and translation. And we talked earlier, maybe last week, I think, about the central dogma. What we've focused on so far is replication. And we talked about the idea that DNA typically comes in this double helix package. The double helix consists of 2 complementary strands, which means that if you know what's on one strand, you know what's on the other strand. If there's an A on one, there will be a T on the other. If there's a C on one, there will be a G on the other. If there's a G on one, there will be a C on the other. And if there's a T on one, there will be an A on the other. It's always paired together in those complementary matches, which means you really only need one. All of the information you need is in one strand. If you have 2 and you pull them apart, you can rebuild the original strand. Find the complementary basis to this one, and you have another version of the original. Find the complementary basis to this one, you have another version of the original. That's how DNA replication works. The problem with DNA is that it contains all the information that your body needs to make proteins. And if that information gets messed up, then you're in trouble. It's like if a recipe gets messed up. If you have a pancake recipe and it's really, really good, and it's got strawberries in it because you love strawberries and you make your famous strawberry pancakes and you slice up the strawberries nice and thin and they're just so delicious and warm and my mouth is watering. But somehow, at some point, somebody gets their hands on your recipe and they're like, strawberries? Sardines. And then for the rest of your life, you're making sardine pancakes and you can't figure out why they don't taste as good. You're following the recipe perfectly and it tastes like crud. Why? Because the recipe got messed up. DNA is the recipe for a protein. Your cells have evolved to keep that recipe safe by taking the DNA and putting it into the nucleus. It's kept inside a relatively secure location in the cell. Cells have evolved to do that because the DNA gets corrupted less because of that. Corrupted DNA makes bad recipes. So keeping the DNA inside the nucleus keeps it from being corrupted by chemicals that might alter it in some way. So that's great for keeping the DNA safe. The problem is, it makes it harder to get at the recipe. If I have my recipe book locked in a vault somewhere, then it's nice and safe, but how do I get the recipe? And the answer is, with DNA, cells have evolved, living organisms have evolved a system for passing a message from the vault of the nucleus to the rest of the cell. And that is through a process called transcription. And what that creates is a new molecule. It's not DNA, but it is a nucleic acid. It's a nucleic acid called RNA, and we'll look at that in a little bit more detail in just a second. RNA is really similar to DNA. There's only a couple of little differences, but the way it's used is very different. DNA is kept secure in the vault. RNA gets passed out of the vault and gets used to actually make proteins. So RNA is used as a messenger, and as a result, some kinds of RNA are called messenger RNA. That RNA is then used to make proteins. The instructions are read by chemicals, molecules, enzymes in the cell, and those enzymes put together amino acids to make proteins. That process is called translation. We're going to look at transcription and translation today. The details of it are basically that we have two complementary strands of DNA. One of those is used to make a strand of messenger RNA, and that messenger RNA is interpreted by a different kind of RNA called transfer RNA that actually is involved with hooking the amino acids together. So the objective, as always, thank you, is to be able to talk about this as an intelligent, reasonably knowledgeable person. Expertise isn't our goal, because no matter what you learn, no matter what you do, there's always going to be a level that you don't get. It's too complicated for any one person to know it all in this environment. But I want you to be able to talk about it. When I start talking about transfer RNA and ribosomes and amino acids, if you have an idea of what that process looks like, you'll be able to follow and you'll be able to contribute to that conversation. So here's what transcription looks like. It creates a strand of nucleic acids, RNA. We'll look at RNA in just a second. It's a little bit different from DNA, but not like tremendously different. But basically what transcription does is it creates a strand of nucleic acid, and that strand of nucleic acid is single-stranded, and it's made out of a slightly different material than DNA. D in the DNA stands for deoxyribo. In RNA, it stands for ribo. So deoxyribonucleic acid and ribonucleic acid are very, very similar, but not exactly the same. We'll look at the differences shortly. The strand of, no, I don't need you to do that. I don't need you to like jump. So the strand of messenger RNA that's created is going to be complementary to one of the DNA strands, which means it's going to be identical to the other one, because the other DNA strand was already complementary to one of the strands of DNA. So this is where the flip-flop starts coming in. So if we have one strand of DNA and another strand of DNA, if the second strand is used to make the messenger RNA, then the messenger RNA is going to be identical to the first strand of RNA. So flip, flop, flip. Does this matter? It's not critical that we think about it in terms of, okay, there's this strand, and then there's this strand, and then there's this strand. What's important is recognizing is that any nucleic acid contains information that can be transmitted by executing a flip-flop. We can make a complementary copy of it, and then make another complementary copy of it, and get the original strand back. RNA is a little bit different because it doesn't have the T nitrogenous base. It has a U instead, and we'll talk about that in just a second. But wherever the original strand of DNA has an A, the complementary messenger RNA will have a U. Wherever the original DNA strand has a C, the complementary strand will have a G. Wherever the original strand has a G, the messenger RNA strand will have a C. And wherever the DNA has a T, the messenger RNA will have an A. So it's the same base pairing rules, except that there is no T in RNA, and we have a U instead. Otherwise, it's the same rules, slightly different substance. Chemically, it's almost exactly identical. It's not exactly the same. So that means if we look at the messenger RNA that was created, it should be identical to the DNA that wasn't used to make the messenger RNA strand. It's going to be complementary to the strand that was used to make it, which means it'll be identical to the strand that wasn't used to make it. Except that everywhere there's a T in the strand of DNA, there's going to be a U in the RNA. So CTAG in this strand, strand 1 of DNA, becomes CUAG in this complementary strand of messenger RNA, okay? And then ultimately this messenger RNA strand is going to be what's used to make a protein. You don't need to know this, but in biology, by convention, we call the strand that's not used to make the messenger RNA the coding strand. This strand has the same information as the messenger RNA. So we say that this is the coding strand. This strand we call the template strand. The template strand has bases that are complementary to both the coding strand and the messenger RNA. So see how it, like the flip-flop is what gets confusing. But if you just think about it in terms of basically these three things are all the same information, it kind of makes sense. It's just a mirror image. So we start with the information, now we have a mirror image of the information, now we have a mirror image of the mirror image of the information, which ends up being the same as the original information, okay? And the only difference is it's a slightly different molecule, but the information is the same. And just like when DNA is replicated, there's an enzyme that does the actual attachment. That enzyme is called RNA polymerase. DNA is put together with DNA polymerase. Polymer is a chain of similar building blocks. Polymerase is an enzyme that builds polymers. RNA polymerase is an enzyme that builds ribonucleic acid polymers. DNA polymerase is an enzyme that builds deoxyribonucleic acid polymers. The vocabulary is kind of insane, but just thinking about the bits and pieces isn't so challenging, okay? We've got 3 mirror images here, 2 of which are the same. All right, let's look at messenger RNA in a little bit more detail. I just want to make sure that you understand that while RNA, ribonucleic acid, is different from DNA, it's not like drastically different. It still consists of a sugar, phosphate, so ribose sugar, phosphate group, and a nitrogenous base hooked together. We still have a sugar phosphate backbone. The structure of the nitrogenous bases is just the same. It all hooks together exactly the same way that your DNA molecules did. But first of all, instead of thymine, we have a slightly different nitrogenous base called uracil. It still attaches to adenine, just like thymine did. It is very, very, very similar, just a little tiny chemical difference. Not so much that it really affects the function, but it's just tiny, tiny bit different, right? It's kind of like using a scrunchie instead of an elastic, right? It does the same thing, it looks the same, but it's not exactly the same. And the other thing about messenger RNA and RNA in general is that it typically occurs as a single strand. So DNA chemically will stick together and make a double helix. So when you have one strand of DNA, it's unstable, but when it's attached to another complementary strand, it's super stable. With RNA, a single strand is actually pretty stable. And that is because of the chemical differences. The ribose sugar and the uracil are what make RNA stable as a single strand. And when we look at the template strand of DNA, a strand of messenger RNA is going to be complementary to that, okay? And here's what it looks like in action. So it's gonna take a sec to get this set up because it has to like load this. But this video is really cool because we looked at another video that was like this, and it shows all this stuff happening in real time, and each little colored dot.
Central dogma of molecular.
Each little colored dot in here represents an atom. So you're gonna see what happens when a strand of messenger RNA is created, okay?
Molecular biology. DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA.
You don't need to know any of that.
The length of DNA following the promoter is a gene.
We'll get into that later.
A recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase.
That you should be familiar with.
It moves the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions.
Don't need to know any of that.
These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand.
Okay.
Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time.
Okay. So, go away. So here's what I want you to see here. Here's the DNA, the double helix twisted up. This is the RNA polymerase, that is the enzyme that builds the messenger RNA. All these little yellow things are RNA nucleotides just floating around in the nucleus of the cell. That's how it actually looks in there. There's just like billions and billions of these little pieces that are just sitting there. If they get close enough to the RNA polymerase, they get kind of sucked in, and if they match, if they're complementary to the base on the template strand of the DNA, then they get attached. This is the strand of messenger RNA that's being created. It's a bunch of RNA nucleotides that have been hooked together by the RNA polymerase.
By matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
That's what it actually looks like, and that's the actual speed of that process. So it's pretty crazy and pretty cool. Okay, so that creates a strand of nucleic acid, ribonucleic acid, called messenger RNA. That messenger RNA then is used to build proteins. The process of building proteins is called translation. Translation is when messenger RNA is interpreted by a different kind of RNA called tRNA. And I'll talk a little bit more about that in just a second. This right here is one piece of tRNA. And it doesn't look exactly like this, but it actually looks pretty similar. One piece of tRNA. And the tRNA works inside ribosomes, which we looked at when we were talking about cells. Ribosomes are the protein makers. Ribosomes work with tRNA to hook together amino acids. So tRNA and ribosomes work together to hook together amino acids to make a protein, right? A protein is also known as a polypeptide. And the way that this works is by using another mirror image. If you look at these 3 transfer RNA bases, they will be complementary to a sequence of 3 messenger RNA bases. They chemically hook together. The A on the messenger RNA will hook to a U on a transfer RNA. The G of messenger RNA will hook to a C of transfer RNA. And the U on the messenger RNA will hook to an A of the transfer RNA. This chunk of transfer RNA will always be associated with a unique amino acid, which means every sequence of 3 messenger RNA bases, nucleotides, will ultimately tell the transfer RNA and the ribosomes which amino acid to put into the sequence. So essentially what's going on is that the messenger RNA is being translated 3 bases at a time. 3 bases stand for one amino acid. And we'll see a little bit more detail on all of this in just a second, and then the work you're going to do today is going to be to practice this, like in groups with pieces of paper and stuff. All right, so let's take a look at translation real quick, and then we'll talk in more detail about transfer RNA, and then we'll kind of put everything together. So this is the same style of animation, and again, it is real time, and
the job of this mRNA.
It's real time, and each little dot is representative of an actual atom, okay?
DNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the 3-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
I love that. Turned into flesh and blood. Like, she goes from just being sort of like calm and composed to like being mega dramatic. And there's some, like, flesh and blood sounds like, I don't know. That's some imagery there. Anyway, so let's look at this. So the animation is great. The downside to the animation is things are moving pretty quickly. There's a lot of words there. So let's take a look at this in a little bit more detail. So here's what transfer RNA looks like. Transfer RNA makes a funny looped shape, most of which we just don't care about. We don't care about this. 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 We care about two parts of the transfer RNA. The first part is the anticodon. This section right down here has 3 RNA nucleotides. They could be any 3, C-U-C in this case, but it could be G-A-A or A-U-U or U-U-U or C-C-C or any combination. We have 4 possible nucleotides for each site. So there's 4 times 4 times 4, or 64 different combinations. Okay, there's 64 different ways that we can arrange 3 nucleotides. So there's 64 of these combinations of 3 RNA letters. On the RNA, on the transfer RNA, we call it an anticodon because it matches with a 3-letter sequence on the messenger RNA that we call the codon. This is the flip again. What's the difference between a codon and an anticodon? What's the difference between the template strand of DNA and the coding strand of DNA? Remembering the order of this isn't key. What's important is recognizing that because nucleic acids have complementary bases, those nucleotides are complementary to each other, you give me 3, and I can make a mirror image, and another mirror image, another mirror image, and another mirror image, and another mirror image, however many I want, without really losing the information. The way it actually looks is like this. We have 2 strands of DNA. One is the coding strand, the other is the template. If the coding strand is ATC, then the template strand is going to be TAG. The messenger RNA that is created from that particular strand is going to be complementary to the template strand. T is going to match with A is going to match with U, G is going to match with C. So we have flipped and then flipped back, and then the tRNA that matches with that will be complementary to that, so this will be UAG. So we flip back again. This is known as a codon. This is known as an anticodon. Coding template will have the same sequence as the messenger RNA codon. The template DNA sequence will match up with the tRNA anticodons. Remembering which is just not as important as understanding that it's basically the same information, right? Like if I show you this, and then I show you this, it's the same information, just 1, 2, 3, 3, 2, 1. And that is basically how DNA and messenger RNA and transfer RNA all work. It's just flip, flop, flip, flop, flip, flop. So the messenger RNA bases that pair with a specific transfer RNA molecule is called a codon. The tRNA bases are called an anticodon. And the other part that we care about is this part right here. This is where there's going to be an amino acid. And each one of these anticodons is paired with a specific amino acid. So if we have a CUC here, it's always going to be attached to the same amino acid, which means if our messenger RNA codon is GAG, it's going to be attached to a tRNA that's attached to a specific amino acid, which means this piece of DNA tells us which amino acids are going to be put together. And we figured all this out experimentally by doing a bunch of chemistry and biology, but basically what it looks like is this. If the messenger RNA starts with an A, and then the next letter is a C, and then the last letter is a G, then A, C, G will always be associated with this particular amino acid. And you do not need to know the names of the amino acids or even the codes for the amino acids, but there's 20 different ones. And it always starts with the same one. So it always starts with an AUG codon, which stands for an amino acid called methionine. That starts the whole process. And there are a few different messenger RNA codons that will cause the whole thing to stop. UAA, UAG, or UDA will all cause a halt. And there's 20 different amino acids. So some of our codons end up coding for the same amino acid. All these codons that start with CC, they all code for proline, which is one of the 20 amino acids. All of the ones that start with GC will code for alanine. And then sometimes it's not as straightforward as that, like the AG starts. There's 2 different amino acids that get coded for, and whether it's SER or ARG is determined by the last letter in the codon. But you can see that if you give me a strand of DNA and tell me whether it's the coding strand or the template strand, I can figure out what the associated messenger RNA codon will be and what the amino acid on the end of the tRNA will be. So all of the information is stored in the DNA, and it gets flip-flop, flip-flop, flip-flopped until we end up with an amino acid. And here's what it all looks like together, okay? We start with a double strand of DNA. One of these will be the coding strand. One of these will be the template strand. The template strand is used to make a complementary sequence of messenger RNA. That messenger RNA will be identical to the coding strand, complementary to the template strand. There, somewhere on that messenger RNA strand, will be a 3-letter sequence that has an A, U, and a G. That's where the whole thing starts. That will attach, a methionine amino acid. That will start with a methionine amino acid, and then every 3 bases along the messenger RNA strand will determine A, what transfer RNA strand is attached, and B, which amino acid gets attached to the protein chain that's forming. The messenger RNA strand will be complementary to the template and identical to the coding strand. The anticodons on the transfer RNA will be complementary to the messenger RNA, identical to the template strand, and complementary to the coding strand. So we flip, flip, flip to get an amino acid. All right, so basically at this point, we've looked at how DNA can be replicated because of its complementary nature, how it can be transcribed to messenger RNA because of its complementary nature, and how it can then be translated into protein with tRNAs because of its complementary nature. The whole point is the structure of DNA determines its 3 key functions: replication, transcription, translation. And what we're going to do now is migrate. So we're going to migrate over to Mr. Barron's room. And so let's do 2 things. 1, take a break. B, pack up all your stuff and migrate to Mr. Barron's room where the work that we're going to do is set up.